Hello, we are Mariona Vidal and myself, Julia Fava, the representative of the Group 107 of Integrated Biomedicine 3. Following on, we are going to expose our project on which we have been developing a research study to elucidate why do children seem to be less infected by the SARS CoV-2. Before starting, let's put ourselves in situation. In December 2019, a novel human coronavirus appeared in Wuhan, China. Since then, the infection escalated to a global pandemic outbreak. The virus has been called SARS CoV-2, and it produces a variable severity of symptoms, from upper respiratory tract infections, nasal congestion, fever, and cough, to more severe symptoms like dyspnea, pneumonia, and even respiratory failure. Recent studies indicate that patients greater than or equal to 60 years old are at higher risk than young age groups, who might be less likely to become infected or, if so, may show milder symptoms or even an asymptomatic infection. In fact, in China, it has been reported that only 2% of patients were under the age of 19. In Madrid region, by March 16, 0.8% of cases were children younger than 18 years old, and data from the United States indicates that among all cases, 1.7% were children or adolescents, and only 3 deaths were registered. Although there is still no major reason that can explain this tendency, there are 2 hypotheses that have a major bibliographic support. On the one hand, some authors point out that the immune system of children and adults are different, both with respect to their composition and functional responsiveness. On the other hand, differences in the location of angiotensin-converting enzyme 2, also known as ACE2, is another possible hypothesis. Due to the current situation of global health emergency, we decided to focus our efforts in this last hypothesis. We believe that this could be a promising research line, since it can be directly carried out in humans and the results could be obtained in a shorter period of time. Therefore, the new data could immediately be applied in other research lines. So, in order to understand it, we have to learn about the virus. SARS-CoV-2 enters to its host cells through the ACE2 receptor thanks to its glycoprotein spikes on the outer surface of the virus, like another 2 coronaviruses that also affect mainly to elderly people. This receptor is important because its location could explain the previously stated symptoms caused by SARS-CoV-2. Moreover, considering previous literature based on in silico models, it is known that ACE2 does not change its activity with age, and neither does change its 3-dimensional structure. This last characteristic only differs between populations since it depends on certain polymorphisms that modify the tertiary and quaternary structure. It has been reported that East Asian populations present a high allele frequency of this polymorphism when compared to European and American populations, and it might be associated with a higher expression of ACE2, which suggests a change in the susceptibility of SARS-CoV-2 infection. So, our main hypothesis is that age-dependent differences in the location of ACE2 leads to a higher expression of the enzyme in the upper respiratory tract of children, and this could be one of the reasons for the observed variation in the symptomatology of SARS-CoV-2 infection. Accordingly, the aim of this project is to quantify the ACE2 expression in adults and children in the upper airways. And to carry out this quantification, a reverse transcription qPCR will be performed. To complement our project, we will also consider the affinity of ACE2 for the spike protein of SARS-CoV-2 in order to verify the dissociation constant observed in silico models. To do so, we will carry out a second experiment using titration ELISA.
Now, let's take a look at how we want to carry out this investigation. This project has been predicted to last for 6 months. If you wish to have more information about the chronogram, we will be pleased to explain it during the questions round. We will select a total of 1,000 participants and we will divide them into groups according to age. We will take people older than 60 years old as one group because we know that they are at higher risk of infection. The second group will be composed by participants from 0 to 19 years old as they tend to show mild or asymptomatic infections. When recruiting the participants, an equitative sex distribution must be taken into account because the ACE2 gene is located in the X chromosome, which means that there could be differences between sex. For further analysis, these same groups will be divided equitably in infected and non-infected after performing the adequate COVID-19 testing. By doing this division, we will be able to determine if infected people have a higher expression of ACE2 or affinity of ACE2 for spike than healthy participants of the same age. Participants who undergo angiotensin converting enzyme inhibitors treatment, common drugs for cardiovascular diseases, and people with comorbidities will be included. These characteristics will be taken as 2 covariants, as they have been related to have implication in the differences in symptoms among ages, but at the same time are very common in our population. which means that excluding them would be unrealistic. Later on, in the statistical analysis, we will determine if, from all the covariants, the difference in AC2 location among age is the one with the most statistical significance and accordingly is linked to the differences in infection. As Julia has explained, there are certain polymorphisms that differ between populations. What made us think that choosing participants of the same ethnicity, in this case Caucasians, would be better in order to obtain reliable results. On the other hand, terminal patients, smoker patients, and pregnant or lactating women would not be contemplated, as the organism's conditions cannot be considered as physiological. Apart from that, people with the inability to consent and or comply with the study protocol will also be excluded. The emergency of the situation asks for quick gathering. Therefore, pooled invasive specimens will be taken through an esophageal brushing. This location is accessible and is the portal of initial infection and transmission of the virus. The process will be carried out twice in each participant. Either RNA or protein will be extracted from these samples in order to perform the RT-qPCR or ELISA technique, respectively. Once we have the samples, we will quantify the AC2 receptor expression through a quantitative reverse transcription PCR. All experiments will be performed in triplicate. First, RNA purity will be controlled by measuring the relative absorbance. Then, the cDNA from the extracted RNA will be obtained and later quantified through a nanodrop. Following on, AC2 and HPRT1 genes will be amplified. The second gene will be used as a housekeeping gene to normalize the mRNA expression levels of AC2. Both expressions will be quantified through a RT-qPCR. The used Dagman primers and probes are commercially available. Finally, the data will be normalized and analyzed. In parallel with the quantification process, an ELISA titration will be carried out in order to verify that there are no differences between the age groups in the affinity of the spike glycoprotein for each cellular receptor. All experiments will be performed in triplicate, and each plate will contain a positive control using a purified AC2 and a negative control. First, protein extracts from brushing samples will be separated equally in two tubes. From the first one, the total AC2 concentration of each sample will be measured through a sandwich ELISA. In this way, the same amount of AC2 from each sample will be taken to do the following step. Once we have done the sandwich ELISA, from the second protein sample we will determine the dissociation constant of the receptor-ligand interaction through a titration ELISA, with a recently described mathematical algorithm. First, a spike protein titration based on serial dilutions will be performed. With this, the right amount of commercially available spike protein will be determined in order to coat plates. Then, the samples will be added and the specific binding of AC2 spike will be formed. After, each well will be incubated with a primary antibody to detect the amount of AC2 bound to the spike protein. Finally, a secondary peroxidase level antibody will be applied to quantify this binding. So the results that we expect to obtain from this project are, on the one hand, to find significant differences in the expression of AC2 in the upper respiratory tract between children, in whom we expect to observe a higher expression, and adults, of which we expect the opposite. This will allow us to contrast our main hypothesis and will also elucidate the importance of AC2 location among the other covariables analyzed. On the other hand, we do not expect to find significant differences in the affinity of AC2 for the spike protein between both age groups, since we believe that the difference in symptomatology lies only on the differential location of AC2 in the airways. Moreover, we do not expect to observe significant differences in any of the two experiments, neither in the AC2 expression or in AC2 spike affinity between infected versus non-infected participants.
We are concerned about the project limitations. For example, the other hypothesis mentioned in the introduction has not been contrasted, and the results cannot be extrapolated to other populations because non-Caucasian participants have been excluded. Also, given the short time in which we plan to collect samples, we may not obtain enough specimens. However, by performing this project, we will contribute to the epidemiological statistics of the studied population by detecting the infection on participants who are or had been infected. Moreover, another project could be carried out following our research line, by instead studying AC2 expression in the lower respiratory tract, which would help to confirm our hypothesis. Finally, the validation of our hypothesis could elucidate new mechanisms for possible treatments, and to decide which path to follow in order to face this pandemic or other coronavirus infections that follow the same age-dependent tendency. We've arrived to the end of the presentation. Because of lack of time, there are details that we have not been able to explain. If you wish to know more about our project, we will be pleased to answer to any question.